Hello everyone, welcome back. In this video, I am going to continue numerical questions series for ICAR JRF plant science exam and also for SRF in genetics and plant breeding and seed science and technology. And this is also useful for all agricultural MSc entrance exam. So this will be the part 3 of numerical questions where I will be taking up the genetics, and there will be a part 4 where I will be covering plant breeding questions. So if you are wanting for next series, like, comment, share and subscribe our channel and encourage us. In this video, to have a clear-cut understanding, stick with me until the end of this video and take a full benefit out of it. Let's get started. Okay, let us start with the first question. So how many types of different gametes will be produced by F1 having the genotype capital A, capital B, small c, small c, small d, capital E, small e, capital F, small f. So they are asking total number of different gametes that will be produced by F1 if we are selfing this combination. So always remember the total number of gametes that are produced by the F1 will be always equal to 2 power n, where n is number of segregating genes. So number of segregating genes or number of heterozygous genes. So you just count the number of heterozygous genes here: 1, 2, 3 and 4. So n value is 4 here. So 2 power 4 is equal to 4 into 4, that is 16. So there are a total of 16 different gametes that will be produced by the F1 with this specific genotype. Next one. So if there are 4 heterozygous loci, the total number of different phenotypes in the F1, if we are assuming that partial dominance at all the loci, will be which of the following. So they are saying that n is equal to 4 and they are asking for total different phenotypes. If in case they have not mentioned this partial dominance, what will be the case? Let us discuss this first. So if it is complete dominance, what will be the formula for finding out the different combinations of phenotype? It is always equal to 2 power n. If in case it was completely dominant. So if we take just an example, if we are crossing tall into dwarf, so we get F1, right? This will be the F1. So if we self this F1, we will be getting, so this is a genotypic combination and the phenotype will be 2 different classes, either tall or dwarf, right? So this is why the general formula will be always equal to 2 power n, since we are getting 2 classes here. But if it is in case of partial or incomplete dominance, we know that if we just cross the F1, let us take it for color now, red into yellow. So if this is the F1, if it is capital R is partially dominant over small r, in that case we are getting 3 different phenotypic classes. Red might be pink or any other color, and second one is lastly it's yellow. So we are clearly getting 3 different phenotypic classes, right? So the formula will be changed into 3 power n. So if they are asking for partial dominance or incomplete dominance, formula will not remain as 2 power n. To find out the different phenotypic classes, it will be 3 power n in this case. So since they have given the n value as 4, it will be 3 power 4. So 3 into 3 into 3 into 3. So it will be equal to 9 into 9, that is 81. So answer would be 81. So just remember, if they are asking for complete dominance, it is always equal to 2 power n, the total number of different phenotypic classes possible. If they are talking about incomplete dominance or partial dominance, this will be 3 power n. Why 3 power n? I already explained this, right? Okay, I hope this is clear. Next one. So which of the following is the percent frequency of capital A B bar small b or small a bar small b? So individuals in the test cross, if there are 10% recombination between A and B, the linkage is in repulsion phase. So they are saying that there has been a test cross, we need to find out the frequency of capital A B, small b or small b, small b. So they are also mentioned that 2 genes are linked in repulsion phase, right? So if they are in the repulsion phase linkage, so it will be capital A small b at the one side, and in the other side it will be small a capital B. So this is how it is. So if we are doing a test cross here, it will be on the recessive parent. So if we are crossing these two, first we need to find the gametes. So what are the two different types of gametes possible? It will be, if we just do the 4 standard double crossover or 4 standard single crossover here, we will just take it as single crossover. So if there is a 4 standard single crossover, there is a total of 4 different gametes that are possible. So first one will be as a parental type, and secondly it will be capital A small a due to the crossing over event, and next it will be small a and small b because of crossing over, and lastly it is of parental type. So out of the 4 gametes that are obtained here, so these two will be called as recombinants. As we know, it is different from the parental type. So last two will be the parental one. So now they have said that there is a 10% recombination. That means the possibility of getting these two recombinants is 10% and the parental is total 90%. So out of 10%, the probability will be 5% for capital A, capital B, and 5% for small b, small b. That is, capital B will have 5% probability and small a, small b will be having 5% probability, or 5% of the gametes will be capital A, capital B, or 5% of the gametes will be small a, small b, and other two it will be 45% of capital A, small b, and 45% of small a, capital B. So this is what they have given. Now we have to find out the frequency of capital B, capital B, capital A, capital B with small a, small b. So what will be the frequency of the gametes? There would be no change. The frequency will be always equal to 1. So there won't be any other possibility here. All the gametes will be small a, small b only. Even if the crossing over is taking place, again the combination that we are getting is small a, small b only. So now we will just have the 5. This is 5 by 100 or 0.05. We can also write it as 0.05. So 0.05, this will be the capital A, capital B, which we just need to multiply with the frequency of obtaining small a, small b. So this is 1. So answer would be 0.05 or 5%. If they have given it in the percentage, it will be, the answer would be 5%, or if they are given it as frequency, it will be 0.05. So answer will be 5% or 0.05. This will be also 5% and this is also 5%. So since it is again 5% here, which is crossed with the other parent, it will be 1. So the answer would be again 5%. Either we are it is same for both, like if it is capital A, capital B bar small a, small b, or small a, small b bar small a, small b, both will be 5%. So answer will be either 5% or 0.05 if they are asking in frequency. Just see the option and just tick the right answer. I hope this is clear. So you have to take care of this repulsion phase linkage. So the reason why I have written the parents in the form of capital A with small b is because they have mentioned that the linkage is repulsion phase. If they are saying it is a coupling phase linkage, the answer will be completely different. So you need to take care or make sure to see this whether it is in the repulsion phase or coupling phase linkage, or and also if they are talking about selfing or test cross. Okay. Next one. So if there is 10% recombination, calculate the percent frequency of capital A, capital B bar capital B, capital A, capital B, or small a, small b bar small a, small b individuals in F2 derived from F1. So this is again similar to the earlier one. The difference is, earlier I have just shown for test cross, now we are doing F2. So what is the difference? Here we will be selfing, we will be not crossing with the recessive parent, we will be just selfing the F1. So F1 would be like, they are clearly shown that this is again the repulsion phase linkage. So that is capital A, small b with small a, capital B. So this is again selfed with the same one, that is capital A, small b and small a, capital B. So they are asking for capital A, capital B with capital A, capital B. This particular combination they are asking the frequency, or small a, small b with small a, small b. So what will be the frequency of it? So we will just form the gametes. So this will be the parental type, secondly it will be recombinant, third is also recombinant, and fourth is again parental. So this is again 10% and last two are 90%. So what is the individual frequency? Capital A, capital B will be 5% and small a, small b will be 5%. So if you just multiply this, so this is 0.05 and this is also 0.05. So they are asking for capital A, capital B with capital A, capital B. So you need to just multiply these two frequency, 0.05 into 0.05. You will get the answer. Just comment the answer on this video, whatever answer you're getting. So if they're asking for capital A, capital B with capital A, capital B, you just need to multiply 0.05 into 0.05, and also same for this one. So if they have mentioned the F1 is in coupling phase, so what will be your answer? If they have mentioned that in the question the F1 is in coupling phase, that is capital A, capital B with small a, small b, which is again selfed. So what will be the frequency of capital A, capital B and capital B, capital B? No. So this will be completely changing based on whether it is in repulsion phase or coupling phase linkage. So if you have understood that, understood these things, whatever I have explained, you just please comment on this video both for coupling phase and also for repulsion phase. Okay. So if you have not understood, you can also comment on this video. I will be explaining this again. Okay. So next one. So in which of the following 4 genes arrangements the greatest number of crossover will be needed to produce the gametes which are capital A, capital B, capital C, capital D and small a, small b, small c, small d gametes. So they are asking for the maximum or the greatest number of crossover that will be required to form the gametes of all the capitals and also smalls. Okay. So just form the crossover here. Maximum crossover in anyhow it will be 3, right? In any case it will be 3 since we have 4 genes here. So just form the crosses in the options itself and see in which of the combination in one strand you will be getting all capital letters, in other strand you should be getting all small letters. So in the first case, if we are doing this cross, we will be getting capital A with small b. So this will be the wrong option, right? Just see the second one. Again, the second one we will be getting capital A with small b. So this is the wrong combination. We do not need it. So third one, it is small a with small b, and next it will be again capital D. So this will be also wrong. So fourth one, just see capital A with capital B, next again with capital C, so next again with capital D. So we are getting completely all the capital letters in the one strand and the small letters in the other strand. So this will be the answer. This will be your answer. So the greatest number of crossover will be required in case of D, that is equal to 3. So 3 is the greatest number of crossovers here. So if you are doing all the 3 crossovers, we will be getting these gametes only in the fourth option. Next one. So if the pollen grain contains 20 picogram of DNA and 8 chromosomes, and what will be the number of chromosomes and the DNA content in the microspore mother cells? So as we know, microspore mother cell is a diploid cell, so it is 2 power n or 2n, sorry. So it is 2n. So pollen grain, we know that it's a haploid and it will be having only n. So if n is equal to 8, 2n should be always obviously equal to 16. So this thing is clear now. So what about the picogram, the DNA content? So again it will be exactly double of in the haploid state. If in the haploid state it is 20 picogram, in the diploid state it will be 40 picogram. So based on this, the answer will be C, 40 picogram with 16 chromosome in microspore mother cells, since it is diploid, where 2n is equal to 16, and also DNA content will be exactly double. Next one. So how many successive mitotic divisions needed to occur in the cell to produce 256 daughter cells? Okay. So this is where most of the students will confuse when they are asking about successive mitotic division. So they are asking about how many generation of mitotic division should be carried out in order to get 256 daughter cells. So they are asking about mitotic division. So it is always 2 power n, because there will be 2 daughter cells that will be formed after each division, right? So how many successive mitotic division should occur, or how many generations of mitotic division should occur in order to get 256 daughter cells? It is simply 2 power n is equal to 256. So how do you write this 256? Again 2 power 8. So n is equal to 8 here. So this would be your answer, or you just need to multiply. So this is 4 into 4, so that would be 16, again 16 into 16, so you will get 256. So like if you do like mitotic crossover for or mitotic division for 8 generations, then you will get 256 cells. This is what it means. So how many successive mitotic division for 8 complete generation? If you do the mitotic division, you will be getting 256 daughter cells. This is what it means. Next one. So find out the number of cells undergoing mitotic division to produce 128 cells. So again, if since they are asking for mitotic division, you need not to do in the similar manner. So you need to read the question carefully. They are asking about how many number of cells will be undergoing mitotic division if it is producing 128 cells. So let us just take a simple example. If one cell it is dividing and giving 2 daughter cells. Let me assume that 2n is equal to 4. Okay, I have got 4 daughter cells. So for getting 4 daughter cells, how many generation like there should be mitotic division? So it is, since 2 power n is equal to 2 power 2, n will be equal to 2. Like for 2 generations there should be mitotic division in order to get 4 daughter cells, right? So this is 1, and again this is second division. So for 2 generation there is a mitotic cross, like mitotic division, so we are getting 4 daughter cells. But observe carefully, for getting 4 daughter cells, how many cells are undergoing mitotic division? So this cell is undergoing mitotic division, this will be the 1, and this is second, so this is also undergoing mitotic division, and this is third, so this is also undergoing mitotic division. None of the others are undergoing mitotic division, because since we have already got the 4 daughter cells here. So for getting 4 daughter cells, how many cells are undergoing mitotic division? It is 1, 2 and 3. So for getting 4 daughter cells, it is 3 cells which is undergoing mitotic division. So we can derive a formula here. It is always like number of cells which is undergoing mitotic division will be always equal to n minus 1, where n is number of cells are produced at the end, number of daughter cells at the end of mitotic division. If it is 128, so that would be equal to n minus 1, where 128 minus 1, so answer will be 127 cells will be undergoing mitotic division to produce 128 cells. So this is what it means. So you need not to confuse with the earlier. Okay, next one, the similar case. So it is again about the division, but here it is meiotic division. So find out the number of meiotic divisions required for the formation of 100 seeds. So to understand this, first we have to know about how the seeds are formed. Seeds are because of pollens plus fusing with the ovary, right? So how the pollens are formed? Pollens are because of pollen mother cell, where because of its mitotic division or meiosis, meiotic event, from the microspore mother cell we will be getting 4 microspores. So which will on the maturation will produce the pollen grains on successive mitosis, right? Based on the successive mitosis we will be getting the matured pollen grain, but initially it is because of the meiosis event where the microspore mother cell will be giving 4 daughter cells, right? That is, one microspore mother cell will give 4 microspores, and this will be getting into 4 matured pollen grains. 4 matured pollen grains. It is simply 1:4 ratio. 1 microspore mother cell is giving 4 pollen grains at the end. So this is what it means simply. So this was the case for pollen. So let us talk of ovary. So megaspore mother cell. So megaspore mother cell will have the one meiosis event where it will form 4 daughter cells. Out of the 4 daughter cells, 3 will degenerate and only one will go into 2 successive mitosis or 3 successive mitosis to give the 8 cell stage, in which there will be 1 egg cell. So if this is the case, every megaspore mother cell is giving 1 egg cell, right? So in order to produce the 100 seeds, there should be 100 meiotic event in case of egg. For egg, it should be 100 meiosis, right? But in case of pollens, to produce 100 pollen grains, only 25 megaspore mother cell should undergo meiosis. So it is 25 meiotic event here. So if we just add these two, it will be 125. So 125 meiotic divisions will give us 100 seeds. So this is how it is. So you should be knowing the concept where to produce the one pollen grain, the one microspore mother, like to produce 4 pollen grains you require 1 megaspore mother cell, sorry, microspore mother cell. But in case of egg cell, for producing one egg cell you require one complete megaspore mother cell, because 3 of which will be degenerating earlier in the earlier stage. Later it is only of the one cell which is undergoing successive mitotic division to produce the 8 cell structure, in which there will be one egg cell. I hope this is clear. So the answer is 125. Next question. So the man with the blood group A marries a woman with the blood group B. Possible blood group of a children is. So they are asking about possible blood group of the children. So it is man with the blood group A. If man is having blood group A, what are the possible alleles here? It can be either IA in the homozygous state, or IA I0 or IO. Okay, so this will be in the man. So woman as B blood group, where again there is a two possibilities of alleles, either homozygous IB or IBIO. So they are asking for possible blood group in case of children. If this is the case, if we are considering either IAIA or IAIO in both the cases for both the parents, so what are the different blood groups or possible blood groups in case of children? So if this is the case, you can get any of this. See, for example, let us take this first one. So if in case it is IAIA in the man, in the woman if it is again IBIO. So if this is the case, we will be getting these 4 different combination. So it can be either capital A, capital B, so that is AB blood group, or again it is AB blood group. So thirdly it is AB again, fourth it is A blood group. So from the first cross, either it can be AB or the A blood group, right? I think third one is BB here, so it is capital B. So either it can be B blood group, A blood group, or AB blood group, or again AB blood group. So this is a possibility from first one. So if you just make out for all other alleles, you will be getting, there is a possibility of getting A, B, AB or O. So make sure to have a clear understanding like how these crosses are made and what are the possible alleles if they are giving the blood group for both man and woman. Okay, so if they are saying that blood group of man is A, so there will be always two possibilities where it will be IA in the homozygous state or IAIO. So you have to consider both the cases or both the possibilities while doing the cross. Okay, if you just consider this, if you just assume both are possible, so you can either get A, B or AB or O. So it will be all of the above. Next one, if the mother has a blood group B, mother is having blood group B. So blood group B means it can be either IB or IBIO, right? So and the son has a blood group A. So if his son is having the blood group A, it can be IAIO or IAIA. So they are asking what will be the possible blood group of father A is. Father is. So if the mother is B, so obviously the father should be having A allele in his blood group, right? So in order to get a blood group of A in case of son, so father and mother, if the blood group is B here or B here, so father should obviously have A alleles to get a son who is having the A blood group. So answer would be A or AB. Even if the father is having IA and IB, still there is a possibility of getting a son with the A blood group. So he should be having, it's compulsory that the father should be having the allele that is IA, and it is even fine if he is having IB along with IA, that is he should be having either A blood group or AB blood group. only in that case the son can have the A group. So the possible blood group will be either AB or just A. I hope this is clear. Next question. So if a character is controlled by 10 pairs of genes, how many different phenotypic classes are present in the population? So observe carefully here, they are saying about polygenic trait. Since a single character is controlled by more than one gene, it is a polygenic trait, or simply it's a quantitative trait. So if it is a qualitative trait, the simple formula is 2 power n, but they are asking about quantitative trait here. So the formula is 2n plus 1, where n is number of genes. So number of genes is 10 here. So it is 2 into 10 plus 1. It will be 21 different phenotypic classes are possible in this case. So make sure to remember this formula. So this is for quantitative trait, where n is the number of genes. Next one. Find the number of AV, light and hybrid DNA containing bacteria if an E. coli with the hybrid DNA is allowed to form a binary fission in the medium containing N14 for 5 different generations. So binary fission is nothing but getting 2 daughter cells from a single bacteria. So this is a mode of reproduction in case of bacteria. So this is simple. So let me talk about the AV, light and hybrid DNA. So AV DNA, what it means? What do you mean by AV DNA? It is simply the DNA is replicated in a medium containing heavy nitrogen, the isotope of nitrogen which is heavy. So it is N15. So N15 is denoted as heavy nitrogen, so heavy isotope of nitrogen. So that is why the name AV DNA. If the E. coli DNA is replicated in a medium containing this AV isotope, the DNA itself is denoted as AV DNA, or it is N15. So what is light DNA? If it is in the N14 media, the isotope of nitrogen, so that is called as light DNA. So what is hybrid DNA then? So if it is in AV DNA, both the strands will be having N15 isotopes. If it is light DNA, both the strands will be having N14 isotopes. So when I talk of hybrid, it is N14 N15. So this is what hybrid means. Okay, so they are saying that for 5 generation there is a binary fission. So at the end of 5 generation, how many cells you will find? So it is simply 2 power n. So it is 2 power 5, which is equal to 32. So total there will be 32 DNAs that will be formed at the end of the cycle. So out of this, it's logically very simple to know like how many will be hybrid and how many will be light and how many will be heavy. So I will just show you the first cycle. Okay, to make it simple, let us consider a single bacterial cell and try to draw a conclusion from it. Okay, so they are saying that single bacterial cell which is having the hybrid strands, that means N14 and N15 strands. So this single DNA is replicated, or the single cell is replicated by binary fission, right? So once it is replicated, again there will be 2 cells. So this is carried out for 5 generations. So what will be the number of hybrid, number of light and number of heavy DNA they are asking us. Okay, so we know that at the end of 5 generation we will be having 32 cells from a single cell. So in the first case, when it is hybrid N15 and N15, so we know the semi-conservative mode of DNA replications, right? So this single strand will be N15. So the progeny strand will be again N14. Why it will be N14? Because it is replicated in the media containing N14 isotope. So the daughter strand will be N14, or the new strand that will be formed will be always N14. So the parental strand, let it be N14, the daughter strand that is formed will be again N14. So now you can clearly see the ratio in the first generation is hybrid is to light. So there is no possibility of heavy DNA. So we'll just cancel this. So possibility is one hybrid and one light. So this is in the case of first generation. Let us take this to the second generation. N15, this one, last one N15. So this is the parental strand. Daughter strand will be again N14 since it is grown in the media containing N14 isotope. This will be the first one, and for the second it is N14 in the parental, and also progeny or the daughter strand will be also N14. So this is again same for the other two. So that is N14 N14 and N14 N14. Now we are getting 4 different cells or 4 different DNAs in which the ratio is 1 hybrid and 3 light. So if you just simply observe the cycle, you are seeing that anyhow you are only getting 1 hybrid at the end of each generation. So there is no possibility that again you will get a new hybrid from the light, since it is purely grown under N14 isotope. So that is why out of 32 cells, 1 will be hybrid and 31 cells will be having light DNA. So this is how you can simply come into conclusion without going into 5 generations. As we know that hybrid is only possible if it is having N15. So N15 is only present in a single strand. So that strand cannot be duplicated. Even if it is replicated, it will be in the N14 media. So that is why every time we are getting the daughter strand as N14 along with the N15. So there will be only single copy even after n number of generation, not even 5. At the n number of generation, the copy number will be single, only one only. That is why out of 32 new DNA that are formed at the end of 5th generation, you will have 1 hybrid is to 31 light DNA, and there is no case of heavy DNA. So this is not at all possible. I hope this is clear. So this is the last question for part 3, and we will be coming up with some more parts. And just now, like I got some questions from one of the students. It was regarding chromosomal mapping. So I'll just try to incorporate those questions in the next upcoming part, and I will just discuss with you all. So again, the same thing, like I would like to repeat the same thing. If you are liking the content, please like, comment, share and subscribe our channel, and so that I will be encouraged to provide more such content. Okay, thank you.